This was a great discovery on his part. Well, we're going to start today on the first series on multi-cell life. Previously, most of the lectures focused on single-celled things, bacteria and archaea, which were really pretty easy to deal with, but to a physicist incredibly complicated. Having learned all that I have come to learn about single-celled creatures, it was really difficult for me to understand how multi-cell creatures could come about. After all, with a single-cell creature, the creature and the cell were one. And that was replication. It was just a simple matter of more or less splitting in two. So how could a genome be developed that somehow could end up allowing the replication of something as complicated as a multi-cell creature? In that case, as I said, the cells are many, and the cells obviously are different from one another. So how do you get a single genome to be able to do that? And besides, there was this timeline that I'm showing you. This isn't the same computer that I've always had. It has a new operating system, which apparently I'm in the process of learning. But at any rate, the arrow, well, I just don't know where it's gone. So try to follow me. That timeline that's shown on the right-hand side of the screen shows that simple cells came into existence in some life form well inside less than 1 billion years after the Earth was formed. And that kind of simple cell life continued on for almost 2.5 billion years. And the appearance of multi-cell life didn't come about until the order of just about 1 billion years ago. And that's a relatively short time. And so, in a way, it seems that you might think of multicellular life as almost being a bigger step than the creation of life itself. Though it's not clear that that is the case at all, because we learned last year from Susan Mahoney that the very close coupling between environment and evolution. And so it could just simply possibly be the case that the delay awaited the right conditions to come along, and presumably that really was much involved with the presence of sufficient and even super sufficient amounts of oxygen. So the focus up to this point has been on the cell structure and function of single-cell creatures, largely just because their cells were simpler and they better illustrated many of the features and functions that of the more complex cells you find in multicellular creatures. But we're of course far more familiar with multicellular creatures because, first of all, we can see them. These single-cell creatures were obviously invisible to us. So differently from these single-cell creatures that we talked about, which remain the same from their birth to whenever they get removed from the environment, we're used to creatures that are born, grow, mature, and die. So while the cells may be similar, the creatures are certainly different. Investigating the cell structure and the function of the cells in those multi-cell creatures will likely uncover for us some surprises. The evidence at a cellular level for the evolution of all life from a single cell common ancestor is compelling. It knits together all living things in ways that we might not have previously anticipated. Some of the material I'll be talking about might be difficult for everybody here to agree with. It will certainly assume a very clean and simple-minded evolutionary point of view. I try to remain open-minded, but it is difficult for me to lead any credence to what's called an AIG point of view. AIG stands for Answers in Genesis. And it's really shocking to learn that only 34% of conservative Republicans, this is 2019, accepted evolution, compared to 83% of liberal Democrats. That's really incredibly disturbing and certainly reflects a failure of our educational system, that these people can go through and become adults and have no acceptance of evolution. So it's clear that at the present time that progress along an evolutionary path required that their transition had to be made from multicellular to multicellular life. Following that 2.5 billion years of single-cell life, the recent appearance of plants and animals within 1 billion years ago, along with the first humanoid showed up just 2 million years ago, and our own species 200,000 years ago. It makes you wonder, is there any acceleration going on? I tried to find that out, and all I could find is that there is a real debate going on about the units that you might measure the acceleration in. All creatures become larger and more complex as the size of their genome increases, along with the number of proteins that they require to keep their various cells functioning. However, the table of how you translate from the nucleotides to amino acids has remained stable over time and across cell types, going all the way down to viruses, which are not even alive. So it ties all living things together in a not necessarily harmonious collection. So there are, as you have been told repeatedly by me, they are those 3 major cell types recognized by biologists: single-cell creatures, bacteria, and archaea, and the multi-cell, which go by the name of eukaryota. The etymology of that word is karyota, is not, and it has to do with the nucleus of a cell that you find in eukaryotes. These guys, the bacteria and archaea, are called pro-eukaryota because they don't have a nucleus to their shell. Now, the eukaryota are multi-cell organisms that as a class have a much larger and complex genome than the single-cell bacteria and archaea. And in eukaryota, the cell is not the organism, it's just simply a part of the thing. I'm really a little lost by my inability to bring up an arrow. Okay, so this plot along the bottom is the size of the number of genomes, the number of base pairs that are in the genome. And if you recall, an E. coli has about 4 million base pairs, and a human has about 3.6 billion base pairs in their genome, and the proteins that those genomes prescribe for, the E. coli has about 4,000 or so proteins that make it up, and humans have the order of 25,000 proteins. And that's what this plot reflects. So here are the multi-celled Uricota at the very top in orange, representing the creatures with the largest genome size and also the greatest number of proteins.
How do you come with that?
Oh my goodness.
How it came on briefly.
Pardon me?
Some dot came on briefly before you changed the slide.
Well, let's see, okay. So let's say a little bit about the chromosome structure of prokaryotes, the single-cell creatures, and eukaryotes, multi-cell. Their chromosomes are really quite different. In prokaryotes, the DNA is a single, very long strand that is circular. It's unbroken, and you remember that you copy it by breaking into the origin and going in both directions, end up being able to make a copy of it. Whereas the eukaryotes have such large genomes that they would be impossible to put them in a circle. So they're broken up into multiple linear strands that are in the form of chromosomes, and there is some ingenious ways that the monstrous genome of eukaryotes end up being wound up, because that genome has to appear in every cell in the creature's body, and wrapping up the genome with, for example, 3 billion base pairs, so that you're able to fit it into every one of the 100 trillion cells you have in your body, is quite remarkable how that's done. We'll come into that actually in the next lecture. The whole issue of in eukaryotes is the transcription will appear inside the nucleus of the cell, and that's because there is so much potential creation that could be going on out of those 3 billion base pairs that the nucleus plays a very big role in actually controlling the rate at which stuff peels out into the body of the cell to be reproduced in ribosomes converted, where the stuff that the RNA that would. So again, this figure that I'm showing, unfortunately, is not really to scale very well. The eukaryotic cell should be the order of 7 times longer, 7 times wider. Overall, it's in terms of volume, a couple of 100 times larger than a periodic cell is. So the eukaryotes that we'll be paying the most attention to are going to be plants and animals. So here is a figure of their 2 cells, and what is striking to you, should be striking to you, is just how similar those 2 cells are. That a plant cell and an animal cell should be so similar in structure. They both have nuclei in the center of the thing. The biggest difference in the 2 cells is the cell wall of a plant is a much stronger thing than the cell wall on animals, largely allowing, for the kind of motion that we need relative to a plant, which doesn't really need that level of fluidity, and also absolutely crucial in the plant cell is that thing called chloroplast, which it turns out all life will end up depending on. In the animal cell, the biggest difference that we have is that thing in red on that side called a centrole. As far as I can see, it's not awfully essential, but it is unique to animal cells, and it allows them to create some of the structures that are needed when the cell is in the process of reproducing itself. So I think the thing to just digest is, first of all, the more or less, what should I say, squared off structure of a plant cell, and the more curvy, fluid structure to an animal cell, and the critical appearance in the plant of the chloroplast, which is going to be able to, by virtue of using sunlight, create all the food that all life on this planet depends on. So the cell division in eukaryotes is more complex than it is in the prokaryotes. If you recall, there the really simple thing was just reproducing its genome, which was a relatively simple thing of just splitting it open, moving in both directions, and recording it. In an animal cell, then, here are the phases that animal cell goes through. There's a phase in which the cell simply gets strong enough to undergo the idea that it's going to replace itself. And then there's a very long phase, especially in these eukaryotes, of copying its DNA, and then it gets itself ready to proceed into mitosis, the process by which the cell breaks into 2 parts. Rather differently from the way it happened in single cells, if you recall, here the chromosome is not just a single piece, it's multiple pieces. They become visible at this phase where you start to be able to see them as colored objects called chromosomes. They're then separated, and here's the role that those centroles have of generating these spindles fibers that come out and align the chromosome at this point, end up then carrying out a separation of those chromosomes. The chromosomes then gather around the spindle, the cell elongates, and finally splits into 2 daughter cells that are presumably a replication of the original cell. And in plants, it's quite similar. They have able again to create spindle fibers, but they do it without the use of any of these centroles, and go through quite a similar path involved in their replication. Again here, just showing the steps that have taken place. We're in a phase now where it's already copied the DNA. Again, it's separation into those 2 ends, the 2 centronomes at either end, and just ending up at the end with a process where it breaks into 2 new cells. Now, in higher eukaryotes, most of that replication is actually done by in a somewhat more complicated and often pleasurable manner. It's not universal, but most reproduction in eukaryotes is typically sexual, involving the union of a sperm from a male and an egg from females. The egg and sperm cells are special cells in that they just contain a single set of chromosomes. So their union together gives you 2 sets of chromosomes. This mode of sexual reproduction has the advantage of producing new combinations of genes, allowing every offspring to be somewhat different from its parent. In spite of the burdens and inefficiencies of sexual reproduction, the diversity in the gene pool that it creates appears to be an overall evolutionary advantage. So below I sort of show the difference between the meiotic replication, where one ends up through a series of steps, just creating 2 of the original types of cells. In the case of meiosis, there is a very different process going on, and it goes through a phase where the genomes are actually end up being mixed, and so the genomes that are produced at the end here are 4 basically just 4 cells with just single chromosomes in them. These should not be just taken as the numbers. There are many of these chromosomes in the case of a human. There would be a total of 46. 22 pairs of them are autosomes, or non-sex chromosomes, and there are 2 sex-determining chromosomes, as written here. The sex chromosomes in humans are called X and Y. Females carry 2 X chromosomes, while a male carries only 1 X and 1 Y. And we'll come back to that in more detail in the next session. But this process of meiosis ends up creating 4 cells that can become either a sperm or an egg, and the uniting of them in sexual union is what begins the life cycle of a eukaryote creature. So you would think that the complexity of a creature would be reflected in the number of base pairs in its genome. I recall that an E. coli has about 4.6 million base pairs, and a human has something over 3 billion, and that prescribes for us about 20,000 proteins, whereas the eukaryote only needs something like 4,000.
Million there should be billion.
Pardon me?
Million, billion? So a million base pairs should be billion, right? Human has 3 point something.
So that's million base pairs.
Or was that 3,000, whatever, 17?
It's 3,000, then it says million in the next line.
17 million, okay, yeah.
Okay.
Yeah. Okay.
So, let's see.
I don't know what the problem is.
Yeah, it's confusing.
Human genome. So one thing that we'll be running into is that in the case of an E. coli, one can understand about 90% of its genome. Let's
It's exciting.
go on. In an E. coli, that simple creature, you could trace and understand that about 90% of its genome prescribed for making the complex molecules that the cell needs to go on. Whereas when you go and look at a human, only about 1 to 2% of that enormous genome is required to make the molecules, protein and RNA type molecules that we need. And so what is what is all the other stuff about? Maybe we're special. How big is the genome of other creatures? And one of the things that to me was just absolutely astounding to see that one of the largest genomes is to be found in flowering plants. There is, I think, a plant called Japanesis something or other that has a cell, a genome so long, so big that it takes it 2 years to copy it. And that, of course, greatly slows down the rate at which you can grow or do anything. When you look at our genome, here, this very little piece here prescribes for proteins, and there are some DNAs. There's a lot of things that are. It's hard to figure out what their purpose is. These introns, these long. Let's see, that's long interdispersed nuclear elements or something like that. So there's just a whole lot of stuff that we don't really understand what its role is in our genome. Here's another map of the genome size with different kinds of creatures. They're all multicellular creatures, so here the genomic size is in millions of base pairs. So here we are at something like 3,000 million base pairs. Out here we're among the things called mammals. We're not certainly the largest, and it is amazing that some of these plants have the largest genome that you could imagine, and what their purpose is not clear. Some of it you might believe is it's kept a history of everything that has attacked it and somehow stored that information away in its genome to be fetched out at some later time. So we find ourselves at this point in the center of a really major uncertainty and a controversy in biology. Some people refer to this extraneous DNA as junk DNA. I think that was a thing that was more fashionable some years ago, and it goes under two different puzzles. One called the C-value enigma, a concern over the lack of correspondence between the genome size and the corresponding creature complexity, and the other is the G-value paradox, the lack of conformity in terms of the number of proteins that are to be prescribed. There's some kind of a little worm that has a genome of 100 million base pairs, but it has 20,000 proteins, as many as we have. It's a complete puzzle as to what this genome in multicell creatures is all about. As I said, there's much about genomes that we don't understand, particularly with regard to eukaryotic creatures. That flower plant, Paris japonica, has 149 billion base pairs, 50 times the size of our genome, and it just takes so long to replicate. Now, you may have already anticipated some of the problems that evolution into multicellular life presents. For example, I have a hard time just understanding how evolution came to handle the issue of reproduction, replication. With single-cell creatures, there's just one type of cell, and you have seen that once the cell's genome has been copied, it's just a matter of cell division for one E. coli to become two. But what about a multi-cell creature? They don't start out as multi-cell creatures. We all start out single-celled, as an egg or a seed. And in that single cell is a complete genome given to us from, or given by the parent to that egg or seed. The fossil record for eggs only is about 300 so million years ago, and seeds seem to occur in the fossil record a little earlier. Somehow evolution came up with a solution of eggs and seeds, or else we wouldn't be here. The parental genome is then, of course, reproduced in every cell of a creature's body. It contains the guidance to direct it through every step. It guides an organism from egg to fetus, to adulthood, and reproductive potential, and to the creature's eventual death. How is all that contained in the genome? Incredible. I feel that we have to stop here and pay a debt to plants, not just because Donna's in the audience, but because there really is an absolute debt to plants to be paid. With photosynthesis, plants are able to just take carbon dioxide, water, available from the air and soil, and use the sun or light as an energy source that allows it to turn that carbon dioxide and water into glucose. In that process, the plant releases oxygen back into the air and stores the energy within glucose molecules in itself. Inside the plant, those small organelles that I showed you, the chloroplasts, they are able to take the energy of sunlight and basically, with that light-absorbing pigment called chlorophyll, is responsible for it's giving the plant its green color. And during photosynthesis, chlorophyll absorbs energy from the red and blue light waves and reflects green. That's what makes the plant appear green. Now, one of the things that I also wanted to point out is that not only do plants, besides providing us with food, they take CO2 out of the atmosphere and put back oxygen. That's a critical role in dealing with climate change. But that oxygen that they produce ends up being absolutely essential in producing the ozone layer, which is, again, absolutely essential to being able to have life on land. Or else the ultraviolet light from the sun would allow to get photons with enough energy they could break up our cells and even break into DNA in a serious way. So there's just an incredible debt to plants. Here I show a picture of some aspects of it. Here's just an oversimplified scheme of what happens with 6 molecules of carbon dioxide with 6 molecules of water. You can put together a single molecule of sugar, and from that also give up 6 molecules of oxygen. The actual chemical process that's going on underneath that simplification is quite extraordinary and very complex, and I don't think that I'm in any particularly good enough shape to go into that at the moment. So here is the issue of cellular respiration. That glucose that's produced by plants likely provided the first food provided the food for the first kind of animal life. I have to believe that that first forms of animal life were herbivores. They were then probably followed by carnivores. But the fossil record is not at all clear on the matter. at the present rate, present time anyway, when you look at how an animal cell breaks down the energy stored in a glucose molecule, it's glycose, a series of incredibly complicated steps called glycosis. I think if I counted them all, there's something like 30 steps to get from glycosis. There's an incredible amount of energy stored in a molecule of glucose. I'm used to units of electron volts for energy, but that's at an atomic level a goodly amount of energy, and that's how much is stored in a molecule of glucose. Your cell couldn't possibly use that directly, so it actually gets it to use that glucose to make 38 conversions of andesine ADP, that's the diphosphate to andesine triphosphate. And that transition, converting this backward, going from ATP to ADP, lets your cell use the energy in small amounts at about 0.3 of an electron volt, which is quite appropriate to what you need for the activities that have to go on in a cell. The remaining 18.6 electron volts out of that 30 just shows up as heat. And when I just first encountered the complexity of this process as it's carried out in humans, I couldn't imagine how it began. How in the name of heaven were you able to do that? And then, just thinking about it for a minute, the plant had already solved that problem, because the plant created glucose, and then it had to be able to take glucose, break it down, and use it to drive its functions. So the ability to do this thing was actually transferred right directly along in the capability of those very earliest cells. Now, just a concluding remark. I, and presumably you, are overwhelmed by the complexity of all this. And there's a current view out there that it's actually far more complex than anything I have really been talking about, because of a codependence of aspects of our evolution that has to be carried out in concert with some forms of microbial life. It's called the hologene genome concept, and it postulates that the holoblast, that's us, the creature plus its symbionts, and its hologenome, the host genome plus its microbiome, is a level of selection in evolution. That had never occurred to me until I recalled the talk given on Saturday morning a couple of months ago by May Gordon regarding the role of microbes in the digestive efficiency of mice. If you recall that talk, she pointed out, or were discovering that mice that were really fat mice were as active and ate no more than the thin mice. But what was happening was they had a genome, they had a gut that basically converted food far more efficiently than that of a normal thin mouse. And she is working on a group that sees this as potentially a way of dealing with world hunger. That is, if you could get people to be far more efficient in their conversion of food into energy and mass, you may have ended up solving a very large problem. And while I could not find out from her in any way whether there was an imprint in the genome of the person due to this particular ability to transcribe food more efficiently, it certainly appears to be a fact. And it will be interesting to see whether taking this far more complex view of how it is that we actually evolved into what we are takes hold. And what she presented was a very hopeful picture. So next time we'll talk about the real problem in multicellular creature. That is the expression of the gene. How does it end up that the genome, which is in your cell, in every cell in your body, knows how to tell each cell that you have to do what it needs to do to keep you intact and present. So, thank you. Oh, so questions?
You could be excused if you don't want to take questions.
Excuse
But
me?
you could be excused if you don't want to take questions.
Oh, no, no.
Since you're literally bleeding.
No.
But do you want to do this, Lorraine? Do you want to do this? Anybody have questions? Paul?
I think I'm missing a part here. The cell, the DNA can reproduce its DNA, a daughter DNA. But why does it go through this complicated process of creating proteins and all this other stuff? Why isn't the world just full of cells or DNA and get rid of people and animals and trees?
Well,
People are,
It seems inefficient, this.
well, there is, I forget who was the author of it, but there is an idea that these things are simply using you as a vehicle.
I feel used.
For hanging around.
Yeah.
You're the one going through all this trouble. They just hang in there and do their thing. What it was called, I think, the something cell?
Selfish gene.
The
shellfish.
selfish gene.
Yeah.
Yeah, exactly. Selfish gene.
Sort of a follow-up to Paul's comment. Evolution retains anything that works. And so if it gets off on a wild streak and it works, it's going to continue.
What does works mean? It just created a big diversion.
No, what works is what reproduces and continues into the future.
You want to go back to primordial ooze?
Yeah, it is funny in many ways. I mean, evolution does seem inefficient. It finds a way that works, and then it just simply sticks with it.
the jury's still out on whether humans.
I think Charles gave me a beautiful example of why you would really question, why didn't evolution come up with a DNA polymerase or an RNA polymerase that could DNA polymerase that could read 3 to 5 rather than doing the very awkward thing that it has to do of making Osaki fragments when it's breaking and trying to recreate the cell of a bacteria. Because it has a thing, it's figured out how to solve this problem, of just having one preliminary that can read in the 5 to 3 direction. And so it gets one leg that it can read beautifully, but the other leg, it has to go through this very awkward stuff of reading in a very complicated fashion.
I just want to make a comment. I just finished reading Randolph Fuller's book on the book that changed America. It's up in our third floor library, and it was done in 2017. And it's all based on the documents that Darwin brought back in looking at his book and his writings, and the transcendentalist of New England, Henry David Thoreau, and I can't remember the professor from Harvard that was involved in all of those. And they're thinking that God put these things on earth where he wanted them. That was sort of how it all came about. And then the argument continued to be argued for a couple hundred years before they eventually accepted Darwin's thinking. So it was a fascinating book, and I thought we're wondering how all this came about now, but 200 years ago they were doing the same question, asking the same questions, and it was just an interesting book.
Huh. Was religion involved in it at all?
Yes.
Yes.
Yes. A lot. And slavery comes into it. The slave issue. They get involved in the slavery issue because of the black population was created differently and had different. So it's a very interesting book.
The author?
Author.
What's the author's name again?
Randall Fuller. And I just returned it. I don't know if I'll go back to the third floor.
I don't know.
How old was the Earth?
It was published in 2017.
No, how old was the world? Do they give an age for the world?
Well, I think it was late 1700s at that time.
Darwin
No.
was 18,
I'm trying to understand how they dealt with evolution. Maybe I wasn't hearing you very well.
Well, they didn't deal with it very well. They were not accepting of it. But eventually Darwin convinced several, and I can't remember the names of one was a professor at Harvard, one was Henry David Thoreau, there was another man that was very involved in the discussion, Alcott. Bronson Alcott was involved. They were giving lectures across the country on the process of evolution and accepting. Okay.
Okay.
I don't know what to do with